Welcome you all. In this video, we are going to see about the fundamentals of crop physiology, CRP 101, and we are going to see I'm going to discuss only about the important topics and the important points that you need to focus during the exam preparations. First, the unit 1. If we come into the unit 1, the first lecture is about the introduction of crop physiology and the importance of crop physiology in agriculture. In this lecture, you can focus on only the importance of, you can see only the importance of crop physiology in agriculture, how the crop physiology is involved in agriculture, like its applied and basic science, and also its importance in nutrient and water absorptions and its role in the yield and crop improvement. Then the second lecture, then coming to the plant cells. In plant, there are several organelles or various types of organelles are found. So and they will discuss about each and individual role of these cell organelles. In this area, you have to focus on how the chloroplasts are involved in the photosynthesis. Likewise, the mitochondria are involved in the respiration process, and some of the cell organelles are double-layered membrane, and some of the cell organelles are single-walled membrane, and some it contains no, it is not membrane bound, and some are unilayer membrane bound, and some are or a half layer membrane bound. So you have to focus on these areas. Then coming to the structure and role of water, you have to read the water potential definitions. That is, the water potential, it deals about the free energy, the capacity of free energy available to do the work. So you study the definition for water potentials and its components. There are 4 components are involved, and you have to see in relation to plant water relationship, only the solute potential and pressure potential are involved. The other 2 are not found in the plant water relationship. So you have to see about the definition of water potential and its components, and also under full turgid conditions and in the partially turgid conditions, the water potential of the plant cell varies, and the water potential of the pure water remains 0, whereas the water potential of any solutions always remains negative. So you have to focus on these areas. And coming to diffusion and osmosis, you have to see the definitions of these diffusion, osmosis, imbibitions and plasmolysis, and the difference between the diffusion and osmosis. The osmosis occurs through the semi-permeable layer. This is a main key important point in osmosis. Then imbibitions and also the plasmolysis. The plasmolysis, it is of 3 types: incipient plasmolysis, evidence, and another one thing. Then you have to focus on this. Then coming to the field capacity and permanent wilting point, here for both field capacity and permanent wilting point, you have to know the difference between what is field capacity and permanent wilting point, and also study the range of the field capacity and permanent wilting point. Then absorption of water, it deals about the water absorption by the roots from the soils, and some of the mechanisms are involved. One is active absorption and another one is passive absorptions. So you have to study both the mechanisms, that is active passive mechanisms involved in water absorptions. In active, 2 theories are involved. One is osmosis, another one is non-osmosis. So you concentrate. The osmosis is based on osmosis. The water is absorbed from the soil by the roots. The non-osmosis deals about against the osmotic gradient. The water is absorbed by spending some energy. So see the difference of the mechanism of water absorptions. Then coming to the pathways of water movement, you see how it is absorbed from the how the water moves from the soil to the xylem of the plant. Then the symplast and apoplast. The symplast deals with the water moves through plasmodesmatal connections, that is symplast. The apoplast, the water or any solutions that moves through the intracellular spaces. It is called as apoplast. So see the definition of apoplast and symplast. Then coming to the translocation of water. The translocation of water, in this translocation of water, you have to see the ascent of sap. Ascent of sap is nothing but the xylem, water moves in the xylem cells. Here also it is here the key notes are one is ascent of sap is otherwise called as xylem movement of the water transport, and it moves only in unidirectional movement. Then its mechanisms. Also here also the physical and vital theory are involved in the ascent of sap. So we focus on this mechanism also. Then coming to transpiration and stomatal physiology. The transpiration and stomatal physiology, you have to see what is the difference between the transpiration and the evaporation. Transpiration is a physiological process, whereas evaporation is it's a normal physical process. So see the difference between transpiration and the evaporations. Then coming to the stomatal physiology, the stomata is made up of God cells, and it consists of nucleus, chloroplast, etc. So focus on the structure of stomata and try to figure out what is the how will be the stomata looks when it is open and is closed. Then the structure of the stomatal pore, it also comes under the stomatal physiology. Then mechanism of stomatal opening and closing. So there are three vital theories are involved in the stomatal opening and closing. Just see about those three theories. Then gutations. Gutations are also gutations. It is occurs watery substance comes out, that is oozes out from the leaf margins, that is called as gutations. See the difference between the gutations and transpiration also. It is an important topic. Then coming to anti-transference. So transpiration is a necessary evil, because if the we can't reduce the transpirations, and most of the 90 to 95% of the water loss is occurs to the transpirations. So to minimize the transpiration, we will go for anti-transference. In anti-transference, there are three types of anti-transference are involved. So see the examples of these anti-transference and the anti-transference, and what are the types are involved. Coming to the unit 2, it is the mineral nutrients of plants. In mineral nutrients of the plants, there are some definitions, or you have to focus on some definitions, and also this is the important one. It is the criteria for essentiality for the mineral nutrients. There will be 3 points. So please go through those 3 points. Then the classification of nutrients. The nutrients are classified into macronutrients, micronutrients, and also the macronutrients, micronutrients, and beneficial. And some of the elements are beneficial elements. So you have to memorize macronutrients like nitrogen, phosphorus, and potassium. Likewise, you have to see all the elements which comes under the micronutrients. Then you have to see what are the physiological functions of the macro and micronutrients, and the deficiency symptoms or also you have to focus. Then coming to the macro and micronutrients, in some of the macronutrients are mobile in plants, and some of the micronutrients are immobile in plants. So you have to clearly know about what are the micronutrients and which are the mobile and immobile nutrients, and its physiological roles, physiological functions. For each and individual nutrients, you have to study the physiological functions and its deficiency symptoms. Then you have to also focus on the corrective measures of those macro and micronutrients. Then another one is the beneficial elements, that which is not involved in the plant. It's supposed not involved in the plant. The beneficial elements are classified as that is not involved in the plant growth and development, and also it does not satisfies the criteria of essentiality. There are some elements like silicon, selenium, they comes under the beneficial elements. So for beneficial elements also you have to note on what are the elements that comes under the beneficial elements, and also for the and those physiological functions and deficiency symptoms. You have to focus on that area also. Next, the nutrients are there are some mechanisms are involved for uptake of these nutrients. So you have to go through the mechanisms. Then hidden anchor. It is for this we can see one graph. They will give one graph. So that is enough. You have to draw a graph for this. So it is also an important question. Then foliar nutrition. The foliar nutrition definitions for foliar nutrition: root feeding, fertigation, stand culture, hydroponics and aeroponics. You have to know what is what. That is the definition of these areas for the for this unit 2. In unit 3, we can see in unit 3 they will discuss about what is photosynthesis, respirations. Mainly they will discuss about the photosynthesis and respirations. The photosynthesis, there will be 2 types. One is light and dark. You have to know what is light reactions and what is dark reactions. The light reactions deals with the photosystem, that is the photophosphorylations. So in light reaction you have to see in which organ the light reaction takes place. Mostly in chloroplast, the photosynthesis happens in the chloroplast. And you have to know what is grana, thylakoid, what are the structures are present in the chloroplast, etc. You have to clearly know about that. And you have to know what is photophosphorylations, and also the cycles of the that is the Z scheme. That is, in light reaction it will be of 2 types. One is cyclic photophosphorylation, another one is non-cyclic photophosphorylations. The Z scheme is also called as non-cyclic photophosphorylations. So you have to focus on both the cyclic and non-cyclic photophosphorilation and the difference between the cyclic and non-cyclic photophosphorilation, and how many ATP and NADPH are produced, and what are the assimilatory powers are produced in the light reactions. Then coming to the photosystems, in light reaction there will be 2 photosystems. One is PS1 and PS2. Each photosystem absorbs different radiation energy, so you have to study those part also. Then the red drop and immersion enhancement effect. In red drop, you have to clearly note down the definition of the red drop and immersion enhancement effect. That is, the red drop happens at the wavelength of if the wavelength is more than 680 nanometer, the short decrease was observed. So that you have to clearly note that at which nanometer the red drop happens and which nanometer the immersion and enhancement effect was take place. So you clearly note down the nanometers and the definitions for the red drop and the immersion enhancement effect. And coming to the photolysis of water, the photolysis of water happens. You have to know how much amount of water enters and how much of electron is released, how much amount of electron, hydrogen and oxygen is produced per amount of water entered into the cell. And also some of the mineral nutrients are involved for the photolysis of water, like Mn and Cl minus. So you have to clearly know what is photolysis of water. And also the photophosphorylations, go through the definition of photophosphorylations and see about the Z scheme cycles. So these are the light reaction process. Coming to the dark reactions, the dark reaction is also called as stromal reactions. That you have to know in which place the light reaction happens and in which place, that is in chloroplast, in which area the dark reaction takes place. In stromal reactions, it is classified into C3, C4, CAM plants. For these plants, you have to know about the photosynthetic pathway, that is the cycle of C3, C4 and CAM plants, and also the how much amount of assimilatory powers are produced in C3, C4 and CAM plants, and also the difference between the C3, C4 and CAM plants. Mostly the CAM plants occurs during the CO2 fixation happens during the night time. The stomata opens during the night time. So you have to clearly know what is C3, C4 and CAMP, its cycles and the assimilatory powers produced. Then coming to the and some of the and the factors that affect during these photosynthetic process is also like sunlight, temperature can affect these photosynthetic process. So you have to know the list of what are the factors that affects both the internal and external factors that affect the photosynthesis. Then photorespiration, it is a major it is also a major part. The photorespiration happens only in the C3 plants, not in C4 plants. You have to know the difference between C3 and C4. In C3, we can see the crans anatomy in C4 plants, whereas the crans anatomy is absent in the C3 plants. So the photorespiration happens only in C3 plants. So you have to know the difference of these two. And the photorespiration is also called the C2 cycle. You have to know all the names of those cycles. The C3 is also called as you have to know what is the other name of C3 plants, C4 plants, CAM plants and photorespiration process. Then you have to know the cycle of the C2 plants and the organelles in which the photorespiration takes place. And in photorespiration, the hydrogen peroxide H2O2 was produced, and this hydrogen peroxide is very toxic to the plant cells. So immediately the catalase enzymes that is present in the photorespirations can convert the H2 hydrogen peroxide into hydrogen and water. Water into oxygen. So you have to know what is photorespiration, what are the enzymes and which are the organelles involved in the photorespiration process and its significance, importance in the plant cells. Then coming to the phloem transport, most of the food materials that are produced in the plant are transported through the phloem. So you have to know what is phloem transport and also the Munch hypothesis, that is the mass flow of what food materials are transported through the phloem. You have to know there will be a diagram. So you have to know what is you have to draw the diagram and what you have to know what is Munch hypothesis. Then the phloem loading and unloading, you have to know the definition. The phloem loading is we can say that the phloem loading is from the source food materials that is produced in the source tissues are loaded into the phloem, and the unloading is after reaching the destinated place. The food materials that are transported through the phloem is unloaded to the sink tissues. First you have to know what is source cells and what is the sink tissues, and for the examples, source cells are mostly leaves, but the sink tissues are young developing leaves of buds, and also the storage organs like fruits, tubers, etc. So you have to know what is source, sink and what is phloem loading, unloading and its example, and the source and sink strength and their manipulations. Then coming to the second part, that is the respirations. In respirations there will be three important cycles. One is the glycolysis, TCA cycle and electron transport chain. For this also you have to concentrate on the cycles and how much ATP and NADH are utilized and how much it is produced, and how the glycolysis, that is the pyruvic acids, is entered into the TCA cycles, and the intermediate steps, that is pyruvic acid is converted into acetyl CoA, and how this enters into the and ultimately the assimilatory powers produced or enters into the electron transport chain, and the electron transport chain converts these into the ATP that is utilized for the further metabolomic process. You have to know what is all these things. Then you have to know what is the difference between the oxidative phosphorylation and the photophosphorylations. This is the major difference. You have to know this is the major questions they will ask about the photophosphorylation and oxidative phosphorylations, and the energy budgeting calculations, and also the respiratory quotient for the carbohydrates, fats and the organic substance. It will be less than 1, more than 1, and for some of it will be act as a 0. So you have to know what is the respiratory quotient and what is the respiratory quotient for carbohydrates, fats, organic substance, protein, etc. Then another one, and the third part is the fat metabolism, fatty acid synthesis and breakdown. Here also you have to know what are the organelles are involved in the plant fatty acid synthesis, and where are the and what are the organelles involved in the fatty acid breakdown, and the important enzymes and its cycle. In Unit 4, they deals about the three important things. One is the plant growth regulators, and another one is the physiology of flowering, that is the photoperiodism, phytochrome, and also the third one is the related to the germination and dormancy of the seeds. So you have to know, first coming to the plant growth regulators. First you have to know what is the plant growth regulators, and you have to know what is the definition of the plant growth regulators. In plant growth regulators, there are auxins. It includes auxin, GA, cytokinin, ethylene, ABA, and also some of the novel and newly generation PGRs like plasmasteroids, jasmonic acid, salicylic acid, and all are also being found. So you have to know what is the what is plant growth regulators, and you have to know all these plant growth regulators physiological role in plants and its agricultural uses in the plants, agricultural uses, that is the commercial uses, and also you have to know the biosynthetic pathway, mainly the biosynthetic pathway, what is the precursor for auxin, GA, cytokinin, ethylene, ABA, plasmasteroids. For every hormones you have to know what is its precursor, in where it occurs, where is the biosynthetic pathway occurs, then which organelle is involved, then what is the what are the various types of organelles are involved in this biosynthetic pathway, and also the if we take auxin, what is the role of auxin? Auxin involves in the root formation, whereas cytokinin is involves in the shoot formations, and also we can see the apical dominance in the auxins, whereas the apical dominance is breakdown by cytokinin, and GA involves in the elongation of the elongation process. So the ethylene, it acts ethylene and ABA for each and every hormone, plasmasteroid, salicylic, for each and every hormone you have to know what are the physiological roles and what are the major you have to know what each and every point of these hormones physiological roles and its agricultural commercial uses in the day-to-day life. And some of the growth retardants that is used for GA, there will be triple C. So you have to know what are the growth retardants and some of the examples for the growth retardants. Then coming to the second part is the photoperiodism. We can see that there are three types. One is short, long, and day neutral plants. You have to know what is short day plant, long day plant, and day neutral plants, and the examples for these three plants. And who discovered the photoperiodism, that is Garner and Allard. Next thing is if we give continuous long light or continuous short light, whether the flowering will happen or not. If there is the long light is interrupted by a short period of light, whether the plant flowers or not in short, long day and day neutral plants. You have to go through like these are the major important points that you have to focus on this photoperiodism. Then Chilochan theory of flowering, you have to know what is this theory and clearly see about what is this theory. It is an important one. Then the forms of phytochrome. The phytochrome is of two types. One is PR and PFR. Forms of phytochrome is one is PR and PFR. The PR observes around 660 nanometer, whereas PFR observes around the 730 nanometer. And the structure of the phytochrome, that is composed of chromophore and apoprotein. And how it regulates the flowering process. You have to know this is the important, that is how the PR and PF4 that regulates the flowering. Like short and long day plants, the phytochrome is also regulates the flowering process. And you have to know what is vernalization, who discovered this vernalizations. Then there will be a theories of vernalizations, that is Lysenko theories. You have to focus on these areas. Mainly in these areas you have to focus what will happen to the flowering process if we flowering process if we change or interrupt the critical period of the flowering. Then second one is the germination and dormancy process. We have to know if for seed germination, what are the physiological and biochemical changes that happens in the seed if the seed is started to germinate. And also in seed dormancy, there are different types. The dormancy occurs due to various reasons, and we have to know what are the methods involved to break down these dormancies, that is breaking methods. And another one, and coming to the another part, is the senescence and absorptions. Senescence and absorptions, which hormone influences the senescence and absorption process, and what are the biochemical and physiological process that is involved during the senescence and absorption of the petioles. And coming to the physiology of fruit, it is of two types. One is climatic and non-climatic fruit. The climatic, you have to concentrate on these things, that is climatic and non-climatic fruits, and the examples of the climatic and non-climatic fruit. And you have to know the difference between what is climatic and non-climatic fruits. It is a major, it is important one. Then what are the factors affecting during the ripening process? That it is break down the cellulose by using the cellulase enzyme, and it involves the pectinase enzyme, and you have to know what are the enzymes involved during the ripening process of the fruits. Then manipulations. We have to see about what are the physiological aspects of growth and development of major crops like rice, maize and sorghum. You have to go through like these crops, what are its growth analysis and its role of and some growth analysis parameters are involved. So you have to see about what is what. Then in unit 5, it deals about two major process. One is the stress physiology, that is the stress. Then another one is the global warming, that is the day-to-day life we are facing the global warming. So coming to the first part, that is the stress. In stress, there are 3 to 4 major stresses are there. One is drought, and second one is the high temperature stress, that is high and low temperature stress, and third one is the salinity, and fourth one is the flooding and UV radiations. But all these stress are very important. In this area you have to focus you have to know what is stress and what are the you have to know what are the types of stress occurs in the plant. Then you have to know if drought occurs in the plants, what are the physiological changes you observe at photosynthetic, that is physiological elements. You have to know what due to drought stress, what are the factors that affects the photosynthetic process, respiration process, and also the stomatal opening and closing process. Here we know the drought, it induces the AV accumulation that will ultimately result in the closure of stomata, so we can reduce the transpirational loss, water loss. So like that a major point is involved. Then for flooding, an Arenheimer formation is formed by causing the death of tissues. For a high and low temperature, you can see some of the photosystems are affected due to the high and low temperature stress. So for each and every stress, you have to know its physiological change, what is the effect of stress at the whole plant level, physiological level, biochemical level, and also at the yield level, and what are the adaptation strategies are involved to manage these stress conditions. For example, in drought resistant, drought avoidance, drought postponement are involved. So these are the adaptations. For salinity, you can see salt overly sensitive mechanisms are involved, and also the salt exclusion, salt compartmentalization of salts. These are the adaptations that the plant will undergo for due to these various types of stress. So there will be compatible osmolytes. What is meant by compatible osmolytes, and what are the osmolytes are involved to maintain the plant water status of the plant, and also the membrane properties, that is a compartmentalization. It happens mainly in the salinity stress. Then how to alleviate these stress? These are the areas you have to focus in the unit 5. Then coming to the global warming, you have in this area you just see what is global warming, what is greenhouse gases, its definition, and due to some of the gases like methane and another due to elevated temperature, elevated carbon dioxide, what are the physiological effects that occurs in the crops, and how these gases affect the physiological process at whole plant level, organ level. These are the major points you have to focus in these areas, and you have to see what is carbon sequestration and how it will how you can sequester this carbon in the soil or ocean. You have to see what is what. These are the So these are the major areas you have to mainly concentrate. So all the best for your examinations. Thank you.